So welcome everybody in our audience, old and new, to the 2024 Machine Learning for Protein Engineering seminar series. We're super excited to get going again and have a lot of awesome speakers lined up for you guys. So today we're kicking it off with Nabil Ibtehaz. He is a third-year PhD student at Purdue in the computer science department and works for Dr. Kihara. And today he's going to be talking about his Domain-PFP model. So whenever you're ready, Nabil, we're super excited. Thank you so much.
Thank you, Meg. Hi, everyone. I'm Nabil Ibtehaz. I'm going to present our work titled Domain-PFP allows protein function prediction using functionally aware domain embedding representations. Thank you, everyone, for joining. All right, so I will briefly go through backgrounds on protein functions and some relevant stuff. We all know that proteins are the building blocks of life, and they kind of govern all the activities in living organisms. Therefore, the functions that protein performs are quite diverse. So they catalyze various biochemical reactions, for example, hydrolyze, ligase, integrase, those kind of activities. They're also involved in transmitting signals, transporting nutrients, and most importantly, they provide structural support by developing extracellular matrix, plasma membrane, and so on. Now, next to all the functions, we have some tokens shown as go, then some number. These are actually called gene ontology IDs, and they're just a standardized controlled vocabulary used in bioinformatics to describe the functions of genes and gene products such as proteins. And gene ontology consists of three main categories: molecular function, which expresses the exact function that is performed at molecular level; biological process, which is kind of an extended version that involves multiple sub-processes; and cellular component represents the location or structures where the protein is active or that contributes to building the structure. And the gene ontology terms have various kind of hierarchical relation between them. For example, they can be part of another function. They can be is a replaced by, or even positive or negatively regulate other functions. For example, for this particular function, peptidase activity, this is also a hydrolysis activity, and this is actually a catalytic activity, and this falls under the molecular function types of protein function. So gene ontology terms are traditionally represented in a directed acyclic graph or in a tree kind of fashion. And this plot was generated using Navigo, which is another project our lab previously worked on. Since protein functions are important, it has been a decade-long problem in bioinformatics to properly predict them. And in order to predict functions, there are various approaches. We all know that proteins that are similar in sequence have the similar structure and function. So sequence similarity is one way to predict protein functions. For example, PFP, which is a kind of a classical method for protein function prediction, was based on sequence similarity. Then protein-protein interaction can also help us identify functions because proteins that interacts together are often involved in a shared task together. And deep graph Go is one particular example of a method that relies on protein-protein interaction. Again, we can predict functions from structural similarity because proteins that are similar in structures, they can bind similar kind of complexes, drugs, and so on. And deep fry is one particular example of a function prediction method using structural information. And for our today's focus is protein domains. And as an example method, we have DeepGo0. So what are protein domains? By definition, protein domains are the structural and functional units of proteins. They are responsible for the particular function or interaction that protein will do, ultimately they control the overall role of a protein in an organism. And one particular protein can have multiple domains. For example, this protein has two domains. One domain that is colored in blue is the IPR276, which is G protein coupled receptor, rhodopsin-like, and it performs the function Go 4930, which is general G protein coupled receptor activity. But this protein also has another domain. The one we have here colored in yellow is called IPR1355, and it's a CXC chemokine receptor 1. And this domain performs these functions. One, it performs CXC chemokine receptor activity in general, and also it performs interleukin 8 binding. So ultimately, due to the presence of these domains in this protein performs these three functions. So if we have the information of the domains in a protein, then we have information of the function that is performed by the protein. And in order to understand the domains of protein, we have several databases that has been developed over very decades, like for example, PFAM, Panther, ProCite, CAT. These are all databases of various domains present in proteins. And Interpro is a compilation of all these databases. So if we want to get the domain information of protein, we can basically go to Interpro because it has combined the domains from all these different databases. And Interpro has a tool called Interpro Scan that is basically a sequence search-based approach from which if we give a protein sequence as input, we can get the various domains that are collected from these various databases. And the Interpro Scan tool is completely sequence-based. It performs various levels of sequence search, for example, single motif method, multiple motif method, or even a full alignment method. So it is very convenient for us to use because all we need is the sequence to extract the domains. And since we know that the function of a protein is dominated by the domains in that, so it is motivational for us to map all the domains and the functions they perform. There are several projects that are trying to achieve that. For example, we have PFAM2Go that annotates the various PFAM families to gene ontology terms. And since we are focusing on Interpro due to the vast collection of domains it has, we mostly work with Interpro2Go. So Interpro2Go is an ongoing project that tries to map the Intergo domains to Go terms. And this is performed manually by the Interpro team at EBI. And although this is treated as a computational method, this is very accurate. Like from empirical studies, we observed that it is more than 90% accurate in most of the cases. And this is a simple example of Interpro2Go data. So we have in various rows, say the Interpro domains and the Go terms associated with that Interpro domain. So we can say that one domain can perform multiple functions here. Like for example, the IPR3 domain is performing the Go 3677, Go 3707, Go 8270, Go 6255, also Go 5634 functions. So yeah, we have all the domains, we have all the functions associated with those domains. Therefore, predicting protein function should be very simple. We will have a simple function that will take the protein sequence as input. Then we will extract all the domains running Interpro scan, and from Interpro2Go mapping, we will extract the Go terms and return the Go terms. So predicting protein function is very simple. Therefore, protein function prediction should be a solved problem. But unfortunately, that's not the case. There are some limitations behind this kind of approach. The first issue is that the annotation of Interpro2Go is limited and it's still ongoing. For example, in the current version, we only have 30,000 interpro to go max, whereas there are around 41,000 interpro domains, families, conserved sites, etc. And we have around 26,000 Go terms. So even if we just had one Go term associated with one domain, still this 30K interpro to go is not enough, because previously we just saw that one domain can actually perform multiple functions. So this annotation is very limited. So therefore, if we try to develop a method like what I presented here, it will lack in terms of will provide a set of domains and we will try to predict a set of functions. But the issue is that this kind of data is very sparse and high dimensional. We can just see that our input will be like 41,000 dimensional and output will be 26,000 dimensional. So it will be very difficult for us to train suitable machine learning models with that. So we moved away from the idea of directly predicting Go terms from domains and try to think of it from a different perspective. Instead of directly predicting, let's try to analyze the associative relationships between them. Suppose in our database, we know that 90% of the proteins with the domain D1 performs a function Go1. And also we know that 87% proteins with domain D2 performs a function Go1. And we have another information that 0.5% proteins with the domain D3 performs a function Go1. Now, if we are given this kind of data, we can somewhat be sure that if a protein has D1 and D2 domains, it is very likely that it will perform the Go1 function. But we can't say anything about the proteins with D3 domains because we don't have sufficient data. So probably this kind of hypothesis will work, but we will need to do a lot of manual stuff, and probably it will not be feasible because there are lots of data to study, and if we study manually, there can be bias and other issues. So what we can do is we can go to self-supervised learning. Self-supervised learning is a paradigm of machine learning where a model is trained on a task using the data itself to generate supervisory signals instead of relying on external labels provided by humans. And self-supervised learning is kind of a very, very broad topic. There are various types, categories, and implementation of self-supervised learning, but we decided to just follow the simplest form of self-supervised learning, where the method is a two-stage method. The first step is just solving an auxiliary or pretext classification task using pseudo labels, which help to initialize the model parameters. Next, once we have the model parameters, then we can perform the actual task we want to perform using either supervised or unsupervised learning. And one similar example can be thought from natural language processing, where we have masked language modeling at the first stage to understand the words, understand the language, and then we can have a supervised method to perform specific tasks like question answering or summarization. So we thought of using self-supervised learning to understand the protein domains and the functions. And in order to do that, we need a large database. So we collected the entire SwissProt database, the release of 2022-3, and there were a total of 568,000 protein sequences. And we annotated those domains using InterProScan 5 REST API. 36,000 proteins had no domains, so we discarded them. And we collected the Go annotation from UniProt. We considered both experimental and computational annotations. And in summary, we had a database of 531,000 proteins with 32,000 domains, 8,000 MF terms, almost 22,000 BP terms, and third, almost 4,000 CC terms. So this was our data set, and we want to understand the domain and function and their relation from this data set using self-supervised learning. So our ultimate goal would be to understand how likely it is that a particular function is performed by a protein given that it has a set of domains. So our task kind of reduces to understanding these conditional probabilities between Go and domain. So from definition, the conditional probability of Go J given domain I can be computed from this formula, and these conditional probabilities can be used as the pseudo levels for our self-supervised learning protocol. And in doing so, we will get some representation for the domains and also for the Go term, but we are primarily focusing on the domain representation. Therefore, we can reduce our problem to this. We will try to understand a domain representation. We will try to understand a Go representation. And we want to have a reasonable representation of them so that one bivariate function can take those representations as input, and it can infer the conditional probability that the domain and the Go terms are co-occurring. So from the data, we have these probabilities. So we already have the pseudo labels understood from the data. Now we need to model our embeddings and this bivariate function. And we model this learning using a simple neural network. So in terms of the domain representation, we have a learnable embedding matrix for the domains. And in terms of the Go representation, we have a learnable Go embedding matrix. So from this, we have obtained the representation of both domains and Go terms. Now we need to model this bivariate function, and we can model this bivariate function using a simple shallow neural network. So our model will compute the Hadamard product between their representation, and we will have a single layer of neural network to predict the conditional probabilities. Now there are multiple considerations here. The first thing is that why are we using a shallow neural network? Why are we not using a deep neural network? So this was something we investigated a lot in the initial part of the study. And we found out that if we use a strong, deep, complex neural network here, the conditional probability prediction becomes very accurate, but the domains are not that much functionally informed. This is because if we have a strong neural network here, even if our representations are weak, the neural network itself can perform the heavy lifting and still can be capable of predicting very accurate conditional probabilities. But if we intentionally provide a weak neural network here, our entire protocol needs to optimize the representations more, because this weak neural network needs to understand the conditional probability from this representation, which ultimately forces the representations to be stronger. That was our primary motivation behind taking a architecture like this, where we have a weak neural network. And this kind of follows the standard practice we have in representation learning right now. Like, for example, in masked autoencoder, we have a strong encoder, but a weak decoder. So this is kind of similar to that. And another thing is that instead of just performing cosine similarity and predicting the conditional probability, we thought of taking Hadamard product, and instead of considering the entire correlation, break the correlation across the various dimensions, because the various dimensions are likely to contain different kind of semantic information. So instead of merging all of them, if we can inspect them separately, probably it will give us an overall more distinctive outlook. And also we want that our domain representation is sparse. So we also applied Elon regularization here. Therefore, ultimately, the bivariate function can easily be represented by a one-line equation, since the neural network is very simple. Now we have the model, so we need to train it to understand the functionally aware domain representation. In order to do that, we collected all the proteins from the SwissProt dataset. Then we collected the domains and functions, as previously mentioned, from InterProScan and UniProt respectively. Then from the entire dataset, we compute the conditional probabilities. And once the conditional probabilities has been computed, we train our self-supervised model to be able to predict the conditional probability given the domains and Go terms. And once this training is done, we will obtain a functionally aware domain embedding. In order to train this model, we also need some negative sample, because this will just provide us the conditional probability that are likely. But we also know that there are various functions not performed by domains, so there is no co-occurrence. In order to also consider that, we randomly sampled 1,000 to 2,000 negative examples to train our model. And we train separate models for MF, VP, CC, since it's a practice in function prediction. And once we have the functionally informed domain embedding, during inference, we can just take a protein sequence as input. From InterProScan, we can get the list of domains. And from our functionally aware domain embedding, we can compute the domain embeddings, by averaging which we can obtain the protein embedding. So in our formulation, we represent a protein as the arithmetic average of all the domain embeddings. And from that protein embedding, we can train a supervised model to perform the function prediction task. To assess our representation most critically, we wanted to adopt the simplest classifier there is, which is just a KNN. So if our embedding is very good, then functionally similar proteins will be placed together in an embedding space, and KNN will be able to predict the function easily. So this is our simple KNN formulation. From the neighbors, we pick a protein, then we see that if this particular Go term is present in that protein or not. If it's present, this identity function is 1, otherwise it's 0, and then we just multiply with the Euclidean distance in the vector space, and we also normalize it. So this is just a straightforward KNN for protein function prediction using our embedding. Now we need to validate whether our domain embeddings are functionally informed or not. One easy way to evaluate that would have been to see if the domains that are nearby in the vector space have the similar functions or not. So we define kind of a domain functional similarity between two domains, which will be the Jaccard index between the Go terms of two domains. As Jaccard index is simply intersection over union, and we consider all the Go terms for which this conditional probability is greater than 0.5. Once we have this kind of rough understanding of domain functional similarity, we randomly selected 100,000 pairs of domains and computed their functional similarity relative to the embedding distance. We considered Manhattan distance because in higher dimensional spaces, it is more meaningful than Euclidean distance, because Euclidean distance has a kind of squeezing effect, but Manhattan distance still prevails a longer range. And if we plotted the embedding distance against the functional similarity, we observe negative correlation. So the domains that are nearby in the vector space are functionally more similar than the domains that are further apart. And this strong negative correlation in all the cases kind of validates that our domain embeddings are functionally informed. So if two domains have similar embeddings, we can assume that their functions will also be similar. Although we have validated that the domain representations are functionally aware, we can't directly say that for the protein embeddings, because for protein embeddings, it's kind of an average of multiple domain embeddings. Therefore, in order to assess that, we repeated the same experiment for proteins. So we will have random pairs of proteins. We will compute their functional similarity and observe how that changes with their embedding distance. Now for proteins, we have a lot more functions compared to the functions associated with particular domains. Therefore, Jaccard index may not be a suitable similarity metric, but fortunately, we already have some established functional similarity metrics for proteins, which is the FunSim score. Basically, in a Go DAG, the FunSim score will first compute the relevant similarity score between two Go terms. It is computed in this kind of formulation. So the probability values are just representative of the frequency. The rarer the Go term or the deeper the Go term, the less probability it has that if we randomly pick a Go term, it will be the Go term. Like for example, it is very likely that if we randomly pick a Go term, we will get this, but it is very unlikely that if we randomly pick a Go term, it will be this deep Go term. Therefore, we kind of look at all the ancestors of pair of Go terms, and this is a metric of how deep the ancestor term is. If the ancestor term is very deep, then it is likely that the Go terms are very related to each other. Like for example, for this ancestor, these two Go terms are very related. But if we just consider this very shallow ancestor, even this Go term is kind of related with this. So we want the tightest bound. That's why we use this kind of formulation. And this is another normalizer to normalize how rare it is to randomly pick that Go term so that it gives us some statistical significance. Next, we consider this between all pairs of Go terms between both of the proteins. And from that, we compute one score for the MF, one score for the BP, one score for the CC, and finally we normalize and we have the FunSim score. So the FunSim score is a bit complicated. Ultimately, it provides us the semantic similarity between the set of functions two proteins have. So we did the same for protein embeddings. We randomly selected 1 million pairs of proteins, computed their FunSim score, and computed their embedding distance. Then we plotted them here, and we can see that as our distance increases, the FunSim score sharply decreases. And only for the proteins that are very nearby to each other, the FunSim score is kind of high. So we can say that our protein embeddings are also functionally informed from this finding. And our self-supervised learning is based on contextual learning. So we would also like to see that if it is possible to infer the manually curated interpret-to-go annotation from our embedding. In order to do so, what we did is that we take our trained model, we pass the interpret domain and Go pairs from the interpret-to-go mapping, and then we compute the conditional probability. We have two versions of this experiment. In one version, the orange one, we consider the entire Swiss prot, and in the another version, the blue one, from the Swiss prot, we intentionally remove the pairs that are presented in interpret-to-go annotation. So this is kind of a very adversarial version of this assessment. And this orange version both has the direct probability information and also the contextual probability information, but the blue version only has the contextual probability information because our model has never seen the pair of domain and Go term in a single protein during the training. Now, if we look at the predictions, we can see that for the orange version, where we don't have any restriction, most of the interpretable mappings has been discovered by our model in both MF, VPCC, and as likely in the all categories as well. But in the blue version, we can see that although the BP and CC kind of have a capture version, but MF loses a lot of the prediction. This is because unlike BP or CC, where we have lots of co-occurrence relation, MF is the most precious molecular function, so it requires direct information. Therefore, for the adversarial version, the MF performance was forced, but still it's very good considered that we are only working with the contextual information. Next, we also try to interpret our models learning. For example, we have this particular domain, IPR 690, which is a Martin type zinc figure domain, and it always co-occurs with another domain, the IPR 36236, which is a general zinc figure superfamily. And this domain is always associated with this Go term. So we are also able to infer this Go term from here with 90% probability. And also for this example, we have various domains that are involved in various kind of viral activities. But this Go term, Go 16032, which is the general viral process, biological process, that is absent from our training. So what is happening here is that all these Go terms are kind of related to viral activities. Like this domain is found in RNA virus, this domain is found in DNA virus, this domain alters the membrane permeability, this domain works in virus replication, and from combining all this information, we were able to infer this viral process. So we were able to combine all the information, but in doing so, we don't blur the information. Like for example, this particular domain is also involved in hydrolysis, but that information was not picked from the viral domains, but it was picked from the super family of IPR 9003. So not only we are able to combine the complementary supplementary information, but we are also able to contrast between the different complementary informations. But the issue comes for MF, like for example, the IPR 174 is a general chemokine receptor, and it is a part of the IPR 276 domain, which is present in GPCR proteins. So from this contextual information, we can understand that this domain will also perform some kind of GP receptor activities, but from this information, we can't say that it can perform our chemokine kind of activity. So we have kind of developed we have kind of developed a representation for the proteins, but we need to compare it with protein language models, since now from unsupervised learning from millions of protein sequences, we have very strong embeddings for proteins. And in order to assess our quality on that, we considered the PRO benchmark, which was published in Nature Machine Intelligence recently. This is a functional benchmark analysis on human proteins, and they collected all the human proteins from SwissProt. Then 50% sequence identity cutoff was used, so that the data are very different, and the Go terms were categorized based on two aspects, one based on frequency. If we have 2 to 30 annotations, or 100 to 500 annotations, or more than 1,000 annotations. And similarly, there was another consideration of the specificity of the Go term. Like for example, if the term is shallow or not, like if it's a very common Go term or not, if it's kind of average, that it resides in the second 1/3 depth in the Go graph, or if it's in the deepest layer of the Go graph. So
using this benchmark, we train a different version of our model by discarding all the human proteins because we don't want any like we don't want any kind of a train test mixing. So we discarded all the human proteins and then we assessed our performance in this benchmark. And we can see that in all the cases, we are performing better than the state-of-the-art protein language models. Like PROT-T5XL has 3 billion parameters, but our 10 million parameters domain group probe model can have 2% higher MF score, 6% higher BP and CC scores, and on average, 4% higher score. And we also tried a more restricted version of this training by not only discarding the human proteins, but also discarding proteins that are kind of similar to human proteins, like we perform sequence identity cutoff at multiple levels of 75, 50, 25, and we can observe that although the performance falls, but in all the cases, still we are better than the state-of-the-art protein language models. And this improvement is more apparent for more difficult Go terms. Like for very common Go terms, we can observe that T5 is actually better than us in both AMF and BP and CC. But for very difficult examples like the very deep Go terms that are also rare, we kind of have a 20% improvement, 40% improvement. So the takeaway point is that large-scale protein language models may perform on or even superior and easy Go terms, since they have access to a lot of protein sequence training data and also their very large complex architecture. However, for more specific and difficult cases, there are still scope of improvement from functionally meaningful protein representations or dedicated function prediction methods. And once we have compared with the protein language models, we also tried comparing against actual state-of-the-art function predictors. Now, most state-of-the-art function predictors rely on multiple sources of information. So probably the domain information is not sufficient. So we also mixed BLAST and protein-protein interaction information in our network. In order to compute the BLAST similarity, we used Diamond BLAST, and we collected the string database from protein-protein interaction information. And then we kind of perform an ensemble of them. So we have the functional probability from domain, functional probability from BLAST, functional probability from protein-protein interaction network. And it is possible that for one particular protein PI, we have no match in the interaction network, or we have no match in BLAST search. Therefore, we also have an identity function to ensure that if there is no match, we are not just adding up zeros. So we kind of do a simple ensemble of BLAST and string database as well. And then we compared the state-of-the-art function prediction methods on the NetGo benchmark. The NetGo benchmark adopts a timeline-based approach. The training proteins are all the proteins annotated before December 2018. The validation proteins are up to January 20, and the testing proteins are between February 20 to October 20, which were annotated. And this is statistics of the NetGo benchmark dataset. So for MF, we have 7,000. For BP, we have around 3,000 Go terms. And in terms of number of training proteins, we have from 60 to 90,000 proteins in the training data. We have a small number of 1,000 validation proteins and around 500 test proteins, but not all test proteins have BP or CC terms. And this is a comparison against the state-of-the-art function predictor methods. And we perform this comparison at multiple levels because it's not a fair comparison to compare every method against other, because some method has only sequence information, some method has also BLAST PPA information. So we try to categorize this comparison at multiple levels. For example, we can consider domain as purely sequence-based information. So we can compare our Domain-PFP against DeepGo-CNN, DeepGo0, these two methods, and we can see that our Domain-PFP actually performs better both in terms of Fmax. So from 662, we have 675 for MF. For BP from 396, we have 0.41. For CC, we have from 0.662 to 0.675. And this is also consistent in terms of AUPR and in terms of SMIN. Fmax is the highest possible F score based on various thresholds. AUPR is just the area under the PR curve, and SMIN represents how statistically important or how difficult is our predicted functions. So we can see that the methods that only rely on sequence information, our Domain-PFP is the best one. The same holds if we consider BLAST, like DeepGo Plus and DeepGo0 plus BLAST also have BLAST information, and we are better than them. Like from 655, we are scoring around 674. Then from 432, we have 434. From 675, we have 681. And if we consider PPI, the same case holds true. Like compared to DeepGraphCo, we are lagging a bit behind in MF. This is because PPA information mostly helps in understanding the biological processes, because that is combination of multiple functions. But since MF is very granular and specific function, protein-protein interaction doesn't help that much for those. So for MF, our performance slightly dropped, but for BP, we're better than DeepGraphCo. Also for CC, it kind of dropped because BP is mostly benefited from PPA information. But when we consider all of them like domain, BLAST, and PPA, we achieve the best score or the second best score or the third best score in all the metrics. Only the NetGo2 server is better than us in terms of MF Fmax, but this is a very powerful ensemble method with access to more information than us. They have domain information, KMR information, RNN embedding, protein interaction information, BLAST information, PubMed abstract, and also frequency information of the function. So overall, we perform very good against the state-of-the-art function predictions, predictor methods, just by simply using a KNN method, a KNN classifier. So this is an assessment of how quality our embeddings are. And another interesting thing is that in the NetGo benchmark, we have one protein, O60099, which has a function of Go 6891 intra-Golgi vesicle mediated transport, and no other methods except Domain-PFP was able to predict that in that benchmark. Only we are able to predict that very deep function, and we believe this came from the existence of the domain IPR24662 in that particular protein, which is directly related to this function. Therefore, we believe that being able to integrate domain information in function prediction methods should help a lot in predicting the very deep Go terms. We also evaluated on the Kappa3 benchmark. We trained the model on the Kappa3 training data set, and we performed best in terms of BP and CC, but still the Go leveler method, which was the winner of Kappa3, performs better in terms of ML. And we have made all our source codes public, and it can be found in GitHub. We also have released our models, and the users can download the models and calculate the domain goal associative probabilities. They can extract the functionally aware protein embedding representations, which can be used for other applications. And last but not the least, we can also just predict the protein
we have some customization options for the user, and they can provide various thresholds. They can provide the option of blast or PPI or not, and can predict the protein function using the code released in our GitHub. But if we don't want to run the code, run the models ourselves, we also have a Google Colab release, and in that Google Colab release, we have all the functions that are present in our GitHub release, but in a much user-friendlier way. Like we can provide the protein ID, or we can provide the protein sequence, or we can upload a FASTA file. And we have also options for blast PPI. We can set the thresholds and then finally predict the functions. So Domain-PFP is open source and can be used for further applications. Now, in terms of limitations, we have some limitations. The first limitation is that it depends on the domain annotation from Interpro. So it's not purely sequential. If some domain is absent in Interpro, then we won't be able to predict the functions. And also another limitation is that if there are unknown domains or unknown Go terms, we won't be able to predict the functions. So our future directions is just based on the limitations. Instead of depending on the domain annotation by Interpro, we are trying to figure out a purely sequence-based approach so that we can probably being able to predict the domains directly from the sequence using deep learning. And we don't need the domain, we just need the functional signature. Just how we have extracted the functional signature of the domain, we try to extract the functional signature of the protein sequences. So this is one future direction of our work. And another future direction is being able to perform zero-shot prediction. If we understood the Go hierarchical relation, then it would have been possible to still handle if the Go terms were unknown, if they were not present in our training data set at all. So this was my overall presentation. I am thankful and grateful to my lab mate at Kimera Lab, which is a combination of computer science and biology students. I'm also thankful to my co-authors, Dr. Ibtehaz and Dr. Derive. And I also acknowledge the fundings for this project, which came from NIH and NSF. So this was my overall presentation.
Thank you everyone for your attention. If you have any questions, I'll be happy to address them. And if you have any further queries, then feel free to mail me. Thank you.
Okay, thank you so much for that great presentation. I'll just read out some of the questions in the chat. And then if anyone else would like to ask a question, also feel free to unmute yourself. So Ella asks, why do you only consider 26,000 Go terms? I thought there were about more than 40,000 Go terms.
Yeah, in the gene ontology list, there are around 48,000 Go terms, I think, the last time I saw. But in the SwissProt data set and in the UniProt annotation, we had only 26,000 Go terms. We have a plan to consider all the Tremble data set to be able to like functionally annotate the entire protein universe. We have a plan for that. But for the SwissProt dataset, we only had around 26,000 Go terms.
Okay, great, thanks. Jiang Yang asks, in addition to Go annotation, can this function-aware embedding benefit other protein downstream tasks such as stability, solubility prediction, etc.?
Yeah, I believe it should be helpful. We're currently working to see that if our embedding can be used for other functional related downstream tasks. Yeah, this is also a future direction for us to see for which other applications this representation can be used.
Great, thanks. And Nabil asks, maybe I missed this in your comparison slide, but did you compare your method to any structure-based annotation methods, perhaps using predicted structures to infer Go labels?
Yeah, we have that comparison. We compared against derive and another method called GetGo that is based on AlphaFold prediction instead of PDB structures. Those that was evaluated on a different data set, and those results are in the supplementary materials of our manuscript, but I'm sorry I haven't presented those results here today. But in that data set as well, Domain-PFP was able to outperform them.
Okay, great. If anyone else has any questions, feel free to unmute or leave them in the chat as well.
Yeah, sure, I can go over the architecture of domain embedding. Yeah. So our ultimate goal was to extract some functionally aware representation for domains, and in order to make it a reasonable representation, it should have some relation with representation of Go terms, and they should kind of correlate with how likely the Go term and domain are to coexist, co-occur. So we followed this kind of formulation that we will try to develop a bivariate function that will be able to predict the conditional probability of the co-occurrence of the domain and Go terms. And we model that in a simple neural network like this. In terms of the domain representation, we have a learnable embedding matrix for the domain. So we will provide the domain ID here, and then in the embedding matrix, we'll pick the corresponding row. And similarly, we'll provide a Go ID here. Then from the matrix, we will pick the corresponding embedding for the Go term. And we want the domain to be functionally informed. So there should be some relation in terms of the functional semantic meaning. So what we did is that we computed the Hadamard product between them, which is the pairwise multiplication. We did this because we could have simply just computed cosine similarity between these two embeddings and then try to predict that. But cosine similarity is limiting because it's combining everything together. And Hadamard product can be considered as a decomposed version of cosine similarity, that instead of multiplying everything together, we are multiplying the different dimensions and then trying to apply some nonlinear functions on top of that. So we followed this kind of architecture, and we intentionally kept the neural network shallow so that the heavy weightlifting is performed by the domain representation, not by the neural network. As I've mentioned during the presentation, we observed that if the neural network is deeper and stronger, then although this conditional probability is very accurate, but the domain embedding becomes weaker. And since we are forcing the neural network to be weak and still learn the conditional probabilities, now the domain representation needs to take the heavy workload and extract and compile as much functional information as possible so that the conditional probability becomes accurate at the end. So yeah, this is the overall architecture of the domain embedding. Does that clarify, or do you have any particular questions?
I think it was more clear this time. Thank you.
Thank you.
Great, thanks. And then another question from Nabil. At what depth in the Go tree do Go terms become more informative than the IPR domain labels? IPR terms can be pretty useful for figuring out functional information for unknown sequences.
Yeah. So basically, as deep we go in the Go terms become more specific and they becomes more informative. And another thing is that IPR terms are very useful. Like just looking at the IPR terms, we can predict the functions. For example, the simplest algorithm I propose here, that we'll just take the IPR terms and then just take the interpretable mapping as function. But the issue is that not all functions are still annotated in interpretable mapping. That's why we needed this kind of approach. And I agree that for the IPR terms that are well annotated, IPR term itself is very useful to predict the functions. And we also observed during some analysis that for some particular IPR terms, we don't even need to compute any embedding because all the possible functions are already annotated. So just copying the interpretable Go terms are kind of sufficient for those particular domains and of course the proteins.
Hey, great, thanks. And then a question from Humud, who asks, how does your method perform on protein isoforms? I know that different domain databases such as SCOP, CATH, PFAM, etc., which Interpro pulls from, have different classification systems, which all bias the fold space in various ways.
Yeah, this is a very good question. We haven't experimented with this. We tried looking at moonlighting proteins and saw that for moonlighting proteins it works, but for protein isoforms, unfortunately we haven't done any experiments yet. But thanks for the suggestion. I will look into this in the future direction of this work. Thank you.
Okay, great. Thank you so much once again for the great talk and sticking around for the questions. And also thanks everyone for attending for your interest today. And I hope to see everyone again for the next seminar series in 2 weeks. Okay, thank you.
Thank you everyone for joining.